In our body, everything is controlled and communicated by the nervous system. Even thought, action, and emotions are controlled by it. It communicates through chemical and electrical signals. Such communications are very rapid, specific, and show immediate response. The neuron shown here is myelinated. These segmented coverings are Schwann cells having myelin sheath inside. Myelination of neurons in CNS, brain and spinal cord is done by oligodendrocytes, and in PNS, cranial and spinal nerves, by Schwann cells. A myelinated neuron contains more lipids, less water, and water-soluble substances than an unmyelinated. In the myelinated region, sodium-potassium channels are absent and highly concentrated at the node. In a myelinated neuron, action potential, which includes depolarization and repolarization, is only generated at nodes. This increases the transmission speed of the nerve impulse, 360 km per hour. In unmyelinated neurons, action potentials are generated over the entire length of the axon, so it has less nerve impulse velocity, 1.8 km per hour. Presynaptic neurons make synapses to either the cell body or the dendrite of the postsynaptic neuron. Potassium is the main intracellular positive ion and sodium is the main extracellular positive ion. Distributions of these ions are maintained by sodium-potassium pump. The cytoplasm of neurons contains impermeable negative ions. At resting conditions, membranes are polarized. Nerve impulse arrives at the axon terminal of the presynaptic neuron. Voltage-gated calcium channels in the axon terminal open and the concentration of calcium ions in the cell increases. An increase in the calcium ion concentration causes the fusion of the synaptic vesicles to the membrane and the release of the neurotransmitter in the synaptic cleft through exocytosis. At resting conditions, membranes are polarized and the potential difference is called resting potential, which has a value of minus 70 millivolts. The neurotransmitter binds to the receptor and opens the chemically gated sodium channel on the postsynaptic membrane. Sodium entry into the cell causes membrane depolarization. Such depolarization is locally or graded. Sufficient stimulus from the presynaptic membrane depolarizes to the threshold level. As the threshold potential is achieved, a voltage-gated sodium channel opens in the trigger zone of the axon, and sodium concentration inside the cell increases, generating the action potential at the trigger zone. Soon after the peak of an action potential, voltage-gated sodium channels get closed and voltage-gated potassium channels open, increasing the potassium permeability toward extracellular spaces. As the potassium moves out, the inside of the membrane gets negative once again, thus repolarized. The movement of positive ions produces a local current that travels away from the trigger zone through the cytoplasm of the axon. As this local current reaches the first node, it excites the membrane to its threshold level and generates action potential there, as well as sends the electric current to the next node. In this way, the action potential in myelinated neurons jumps from node to node. This increases the transmission speed of nerve impulses in myelinated neurons, approximately 120 meters per second, whereas in unmyelinated, 0.5 meters per second. Finally, the nerve impulse reaches the axon terminal, and in the same way, the impulse will be transferred to the next neuron or organ.